All right. So good evening, everyone, and welcome to this very first NeuroMeetup talk for 2022. So today's session is going to be dedicated to predictive coding, a very interesting topic, which is going to be of interest for both experimental and theoretical neuroscientists. And today our speaker will be Nicolas Merag. So Nicolas firstly graduated in space engineering from the ISAE Super in France. And he recently obtained his PhD thesis in neuroscience from the MIT, from the lab of Mehrdad Jazayeri. So congrats, Nicolas. And you are now a postdoc fellow at the Institut de Neurosciences de la Timone in Marseille. So I would just like to remind the audience that you are able to ask questions in the chat. After the talk, we will have some time to ask questions and to answer them. And that being said, Nicolas, the floor is yours.
Okay, great. Well, thanks for the short introduction. And so as you mentioned, the talk I'll be presenting is going to be mostly about the work that I've been doing toward the end of my PhD at MIT, working with Mehrdad Jazayeri. And the topic, as you will see, is related to predictive processing in monkey electrophysiology. So as a starting motivation, I wanted to start with a sort of a simple example. Just imagine that you're trying to learn to play baseball. So when you're learning to play baseball, what you can do is you look typically at the ball and you're trying to estimate the speed of the ball in order to know when to bat. So in this particular case, you might be looking at the ball and estimate that it goes somewhere around 40 meters per second, but you're not sure because the ball goes very fast and you're sort of a naive player. So the chances are you will miss the ball. After enough attempts, after sort of collecting enough experiences, now, in addition to estimating the speed of the ball from looking at it, now you have something else that you can use. You can use your prior experience that maybe tells you that this particular player has been throwing on average at 45 meters per second. And so now the idea is that you can use these two informations in order to come up with a better estimate of the speed of the ball. And maybe here you'll estimate that it's somewhere around 42 meters per second. And it may actually help you perform better. So Bayesian theory is a conceptually powerful framework to explain how these two types of informations can be combined. So in the framework of Bayesian theory, the sensory measurements are represented through a distribution called the likelihood distribution. And in our example, the likelihood represents simply the range of speeds that I can potentially measure given the true speed of the ball. On the other hand, I have my prior expectation, which is represented by the prior distribution, which effectively is the range of speeds that I can expect from all my past experiences. And so Bayesian theory gives us a way of combining these two distributions in order to obtain a third distribution called the posterior, from which we can then obtain an optimal sensory estimate. In that case, that's what gives you the 42 meters per second. So the power of Bayesian theory is that it can quantitatively predict how our behavior can become biased towards our prior expectations. And that bias will be a function of the relative uncertainty between our sensory measurements and our prior expectations. So if our prior expectations are highly reliable, which the reliability here is given by the width, how narrow this distribution is, the bias will be large, meaning I'll be highly biased towards my prior expectations. If, however, my prior experiences are less reliable relative to my sensory measurements, now the biases will be much smaller. So with that small introduction about the concept of Bayesian theory, here are the two main questions that I tried to address during my thesis and which I will be talking about during the talk. So the first question is to understand how prior experience in the temporal domain can influence behavior of a subject. And the second question is how are temporal expectations in fact represented in the brain? So I'll be describing the results of two different papers that we published 2 years in a row, and you'll see that the results are highly intertwined between the two papers. But so if you're interested, definitely you can go and dig more deeply into them. So the task that I used during my PhD was the ready, set, go task. And it's effectively a time interval reproduction task in monkeys. So the idea is that we will ask a monkey to measure the time interval between two sets of flashes. So there's the first ready flash, followed by the set flash. And after the set, the monkey has to shift its gaze towards one of the peripheral targets that can appear either on the left or on the right of the screen. So the objective of the monkey is on every trial to measure the time between the ready set, which is called the sample interval here, TS. And then it has to use this sample interval and reproduce it immediately after by waiting the same amount of time before initiating the saccade. So really the objective is to make sure that this produced interval here, TP, matches the sample interval that we present. And to be more specific, the animal is rewarded based on the relative error between the TP and the TS. So if you want to keep a sort of an analogy to help you think about this task, you can think of it as a harder version of baseball where you only see the ball twice. When the ball leaves the glove of the person who throws it, a second time when the ball is midway into the air. And based on these two sensory information, you have to decide when to initiate your batting. So we use this task to manipulate temporal expectations of the monkeys by choosing the sample intervals from two different distributions. So there's the short distribution, color-coded in red, and the long distribution, color-coded in blue. And each of the distribution has 5 equidistant intervals, and the distributions are sampled uniformly across trials. And the idea is that we're going to switch between the two distributions across short blocks of trials. How do we signal to the animal which distribution we use on a given trial? We change the color of the fixation point. So if the color is red, the animal should expect an interval to come from the short distribution. If the fixation is blue, the interval will be coming from the long distribution. Keeping the baseball analogy, we're putting the monkey in a situation where he's facing either a red opponent who throws the ball very fast or a blue opponent who throws the ball relatively slowly. So before showing the behavior of the monkey, let's think of how, in terms of the Bayesian framework, how the monkey should behave in this task. So as I mentioned, the goal of this task is for the monkey to measure the time between ready and set, the TS, and to reproduce it between set and go. So we can consider that on each trial, the monkey is making a noisy measurement of the TS, which is called here TM, and that's represented by this likelihood distribution here. On the other hand, the animal, we can assume that the animal may have learned the distribution of intervals that we tend to present. That is to say, the animal has developed a prior distribution over the intervals and may use it in complement with the sensory measurement. So in other words, these two distributions, the likelihood and the prior distribution, can be combined to get the posterior, from which we get an optimal estimate here, the TE. And finally, the animal may use this sensory estimate and produce a noisy version of it, which is going to be the final TP that we measure or we record experimentally. So now let's consider a specific case where, for instance, we're presenting to the animal the middle interval here. So in that case, the animal might be measuring this specific TM, and the prior distribution around the measured TM is in fact completely uniform. So it is a case where the prior is completely, we call, uninformative. And as a result, the estimate that the animal will come up with will be exactly equal to the measured interval TM. So if now I plot the behavior of the predicted behavior of the animal, on the y-axis is the produced interval, and on the x-axis is the sample interval. For this particular middle interval, we expect the animal to produce TP to be exactly equal to TS. That is to say, this point should be on the unity line. Now, if we consider another case where now the interval is at the edge of the distribution, well, now something a bit different happens. Let's assume that this is the measured interval that the animal makes on this trial. In that case, the prior distribution, the prior expectation can be used to rule out a bunch of values which are sitting on the left of this barrier here. So in other words, when you combine this likelihood distribution with this, the prior, now your posterior will become a truncated version of your likelihood. And the average of the posterior, which you will use as your estimate, is going to be biased towards the middle of the distribution. And so that is what I'm representing here by showing that for this specific TS, we expect the produced interval TP to be biased towards the mean and to be away from the unity line. So if you generalize this to the entire distribution, in the end, what you predict is that if the monkey is using its prior expectation, the response profile should be nonlinear like this and show some behavioral biases towards the mean at the edges of the distribution. So here I'm going to plot actually the behavior of the monkey using the same format. So on the x-axis is the sample interval, and on the y-axis is the produced interval. And the lines here represent the predictions from the Bayesian model. So as you can see, the behavior of the monkey represented by those circles, which show the average response for each interval, are in agreement with the prediction of the Bayesian model. And one case that you can focus on to help you understand what's happening here is if you consider this interval, which is at the overlap between the two distributions. So in this specific case, the interval that we present is exactly the same. However, you can see that the average response of the animal is very different. And in fact, you see that the biases are opposite depending on the prior expectation of the animal. So what these behavioral biases tell us is that animals are indeed relying on their prior expectations to solve this task, which means then that we can go into the brain and look for a potential neural correlate for these behavioral biases. Stop me at any point if there's any questions about the task or before I move on to the physiology.
So far, so good. So you can go on.
Great.
Yeah, I would just like to remind the audience that you can ask questions directly in the chat.
Okay, so if nobody has any questions about the task, so during this task, we went and recorded from an area of the dorsomedial frontal cortex of the monkey, which is an area known to be implicated in the timing of behavior. And in fact, we know in humans, for instance, that patients that have lesions in this area tend to have specific deficits in the time domain. And specifically, they lack the ability to use their prior experience somehow to help them perform specific timing tasks. So based on the literature, we reasoned that this was a good candidate area to go and look for the representation of prior expectations in the time domain. For the electrophysiologist in the crowd, the recording setup that I used was a set of 3 linear probes with 24 channels each, which allow me to acutely record from about 100 neurons on every day on a good day, 100 neurons on a good day. Okay, so now let's look at what single neurons in this task do. And first, I'll be focusing specifically on the neural activity during the measurement epoch. So that is between the ready and the set. On the x-axis will be the time aligned to the ready flash. And on the y-axis is simply the firing rate of the neuron. So here is one example neuron. Now the color code will always be the same throughout the presentation. Red is for the short distribution, blue is for the long, and the different shades of red and blues are for the different intervals within each distribution. And here the circles are simply showing you when exactly the set flash happens for each interval. So here's one example neuron, and now the six example neurons taken from 2 different animals. So the first observation you can see is that neural activity is highly heterogeneous across neurons. However, you can also see that patterns of activity are highly different for some neurons between the 2 conditions, which is in itself remarkable in the sense that the animal is effectively performing the same exact task in both conditions. The animal is simply keeping track of time, but depending on which condition, which distribution is skewed, you see that the dynamics is very different. And we'll come back to this later. Okay, so now let's look at neural activity in the set-go epoch, which is the production epoch. And the format is the same. Now the time along the x-axis is aligned to the set flash, and on the y-axis is the firing rate. Here's one example neuron. And now you see that the patterns of activity are separated now by each interval because this is the production epoch and the animal knows which interval to produce. And here are the 5 remaining neurons. Again, you see a lot of heterogeneity across the population, but you also see some interesting structure that emerges, structure in the sense that those patterns of activity seem to be ordered with respect to the interval that the animal is producing. So because of the heterogeneity across the neurons, one typical approach to coming up with a better sort of visualization of the data is to move on to population level analyses. So here the idea is to summarize the population of your entire population. And here we're talking several hundreds of neurons at a time. You want to summarize this activity in a high dimensional space where each dimension represents the activity of a single neuron. So assume you're looking at the population activity across all your neurons at a single time t. This pattern of activity can be summarized as a state in the space. So if you project this particular state along each dimension, you will recover the population, the firing rates of each of those individual neurons. Now, if you consider a different time point, now you move to a different state. And if you span the entire epoch, now you recover what is called a neural trajectory, which is simply a collection of states across times. Okay, so now I'm going to show you a video that shows you the dynamics, the neural trajectories associated with the 2 different conditions during the ready set epoch. And again, the color will be the same, red for the short and blue for the long. So let's look at the video. So obviously here there's a step that I omitted, which is omitted to say is that we apply some dimensionality reduction technique to be able to visualize this in 3 dimensions, because of course we have several hundreds of neurons and we can't see a video in hundreds dimensions. But if people have questions, I can come back to this. So just to clarify here on the video, the state at the time of ready is somewhere here. And as you could see, the dynamics evolves along this direction and has this interesting curvature here. And these are the different conditions. So each trajectory is one condition, but they are grouped here by condition. Let's look at it for a second time. Okay. And then here is a video of the same thing, but for the set-go epoch. So now you should expect the neural trajectories to be really separated according to the interval and not by the condition, because the animal knows the interval. Okay, so now the activity starts around the time of set here and moves rapidly and makes a turn here and tries to converge to this state here, apparently. Let's look at it one more time. Okay. So here are simply the 2 static pictures of what you just saw in the video. And essentially in the remaining of the talk, what I'll try to do is to explain to you how we were able to relate specific features of the dynamics that you see both in the ready set and the set go epochs to the temporal expectations and the behavioral biases that we observe in the animal's behavior. And first, I'll be focusing on the set go epoch. So for the set go epoch, we didn't start from scratch because many studies before us had studied what happens when you ask an animal to produce intervals of different duration. It's been known for a long time that when an animal is producing a short interval versus a long interval, the neural trajectories seem to be very similar, but the only difference between the two is the speed at which they evolve. So for a short interval, the activity evolves along this trajectory very quickly and reaches this point here, which is you can see it as the action triggering state. Whereas for a long interval, the dynamics will evolve along a very similar trajectory, but simply will move a bit slower in order to reach this state at a later time point. And that's what supposedly allows the animal to control the time of the movement initiation. Then the second thing that we knew from the literature is that what determines the speed of the trajectories is in fact the state, the initial condition of the trajectory, that is to say the state from which it starts. And that is what's shown here, where if your initial condition is located up, then you will move relatively slowly along your trajectory, whereas here, if your initial condition is located at the bottom, your speed will be much faster. So qualitatively, in our data, when we looked at the dynamics during the set go epoch, we observed what we already knew or expected from the literature. We saw different trajectories, which were moving at different speeds, and the speed was faster for intervals that were corresponding to shorter durations. So here again, the red, darker red means very short interval, and this is the trajectories which has the fastest speed. And just to clarify here, the speed, you can infer it from looking at the spacing between the different states along the trajectory. And they are separated by 20 milliseconds. And in addition to the speed modulations, we also noticed that the trajectories were initiated from different states at the time of set. So we quantitatively verified what we expected from the literature. And indeed, we find that the speed of the trajectory was highly dependent on the initial condition at the time of set. And this was the case for the two conditions separately. And second, the speed of the trajectory was highly predictive of the interval that the animal produces for each of the intervals. So in the end, the underlying picture that you should have in your head for what happens in the set go epoch is that at the time of set, the population dynamics is initialized at some states along this dimension here, and the level of the position of the state along this initial condition axis will determine the speed at which it will evolve towards this finish line here, represented by the dash line. And whenever the activity reaches this line, the animal will trigger the movement. So the key question for us was to understand where the biases came from, the biases that I presented in the behavior at the beginning. If we assume that the initial conditions are organized as I'm picturing it currently, we would expect a completely linear profile for the speed. In other words, we should expect, if that is the case, that the produced interval should be completely linear and lie along the unity line. We know it's not the case. We know that the animals are showing some behavioral biases. So we needed to come up with some hypotheses for how those biases might emerge. The first hypothesis we had was that perhaps this finish line in the population state space was warped in a sense that the distance that the trajectories associated with the longer interval needs to travel is a bit shorter compared to the trajectories associated with the shorter intervals. That would lead to the same behavioral biases that we see. A second hypothesis was that it is not the finish line, but the initial condition itself, which is sort of biased in the sense that the initial state could be further along the trajectory only for the longest interval, and could be further up the trajectory for intervals which are shorter. And again, this configuration would give us some biases as we observe in the behavior. Finally, the last hypothesis we came up with was that perhaps the structure of the initial condition along this axis was itself warped. What I mean by that is that the initial condition associated with the shortest and longest interval could be brought towards the initial condition associated with the middle interval, such that now the speeds for those extremes intervals would show some biases, leading to a nonlinear speed profile. So to try and answer or to arbitrate between these different hypotheses, we looked at the dynamics during the preceding epoch, preceding the set, that is to say, during the measurement epoch. And there, what we had observed was that the dynamics seemed to show some interesting curvature, specifically in the range of intervals that the animal is expecting within each of those distributions. So the hypothesis 3, we refined it by saying that perhaps what happens is that during the ready set, the trajectory becomes curved within the range of intervals expected. And that curvature then allows the set of initial conditions to be linearly read out and biased toward the mean, the initial condition associated with the mean. To make a long story short, in the data, we confirmed that this hypothesis 3 was correct, and we were able to reject hypothesis 1 and 2. And we did that by looking at the organization of the initial conditions at the time of set. And we found that these initial conditions were showing the same nonlinear pattern that was predicted quantitatively by the behavior itself. So in the end, here, the full picture that we uncovered is that the Bayesian biases that we observe in the behavior originate from a curvature in the neural trajectory, which is specific to the range of intervals that the animal is expecting. Now, the last piece of observation that we needed to sort of wrap our heads around was the fact that the trajectories during the ready set epoch not only showed this curvature, but also had differences in speed themselves. So we weren't expecting speed differences per se, because the animal during the ready set is not engaged in any form of motor preparation. The animal is simply measuring the time. So to try and interpret those differences in speeds, we came up with a hypothesis which was directly related to the idea of predictive processing. The idea was that, or we hypothesized that the speed was adjusted according to the mean of each distribution, which then would allow each individual interval to be encoded as a deviation away from the mean. So if you look at this schematic here, if the speed of the two trajectories is adjusted according to the mean, then the different intervals here can simply be encoded as a deviation away from this state for both distribution. So this hypothesis was appealing to us because, first of all, it related to the idea of predictive processing, which has been out there for a long time, but also because it makes a highly quantitative prediction about the data that we could just go and test directly. The prediction is that the speed, as I mentioned, the speed along the trajectory should be inversely related to the mean of the distribution. So to test this prediction, I designed an analysis which allowed me to measure the speed differences between the two trajectories. So I did that by asking, how much time do I need to travel a certain distance d along the short trajectory? And I'm going to call this time t short. And the same question, how long do I need to travel the same distance d along the blue trajectory. And this time is called the T long. Here, what I'm plotting is the T long versus T short. If I increase the distance and I start going along the entire trajectory, for each distance, I get a separate point. And in the end, if I cover the entire trajectory, I obtain a mapping between T long and T short. And the reason why this mapping is interesting is because we expect the slope of this mapping to be directly related to the speed differences that are along the trajectories. Specifically, according to our prediction, the slope of this mapping should be exactly equal to the ratio of the two means, the mean of the long distribution over the mean of the short distribution. So now I'm going to plot for you the empirical mapping that I recovered from the data. So plotting the T long versus T short. Here's the unity line. And here the red line is the prediction, which is simply a line whose slope is equal to the ratio of the two means. And so here the black is the data. And as you can see, the data agrees well with the prediction. And this was the case for the two monkeys separately. So from these analyses, what we concluded was that the speed during the ready set epoch quantitatively matched the animal's prior expectations, which was in this case, the average interval expected for each distribution. Okay, so then as a side note, there was an important control experiment that I needed to perform to make sure that the speed differences were truly reflecting differences in expectations and not differences in motor planning. So let me just explain this for a second. So far, what I've hypothesized is that the speed during the ready set reflects the mean of the sample distribution. I'm going to call this hypothesis the expectation hypothesis. There is a second alternative hypothesis, which is that the speed during the ready set could in fact reflect not the mean of the sample distribution, but instead the mean of the produced distribution. And that hypothesis is called the motor planning here. This hypothesis is sort of believable a priori simply because we know that there are speed differences during the set-go epochs, which are related to the interval that the animal needs to produce. So it could be, in theory, that the differences during the ready set come from differences in motor planning. So for this, I used a control experiment in which instead of having two different sample distributions, now I had two identical sample distributions. But in one condition, I asked the monkey to measure that interval and to multiply it by 1, whereas in the second condition, in blue here, I asked the monkey to measure the interval and multiply it by 1.5 and then produce that interval. So this creates a situation where across the two conditions, the temporal expectations is the same. However, in terms of motor planning, the distributions that the animal produces are different. So according to the expectation hypothesis, which is our favorite hypothesis, in this controlled experiment, we should expect the speeds to be equal across the two conditions. And that is because the sample distributions are the same. However, if the motor planning hypothesis is true, in that case, we should expect different speeds because now the distribution of produced intervals are different. So again, to make a long story short, what I found by applying the exact same analysis to the control data was that the speed differences, there was no speed difference between the two conditions. And so here, this is simply the temporal mapping that I used before, but on the control data. And as you can see, this temporal mapping is exactly along the unity line, which you can compare to the original data where we had the data away from the unity line. Okay, so here's a summary of what I described so far. So in this ready, set, go task, what I showed you is that the dynamics during this first epoch is adjusted based on the animal's expectations that is to say the speed reflects the mean interval, the time intervals is then encoded along a curved trajectory which is centered around the mean. This curvature allows the initial conditions here, the initial states, to be biased toward the state associated with the mean. Then this warped representation of the initial conditions allows the speed profile to have some bias. And finally, this bias speed profile leads to the biases we see in the produced interval. Okay. So any questions so far before I finish on the last part of the talk?
I think it's still all good. You can go on and we will have questions at the end of the talk.
Okay, no problem. Okay, so for the last part of the talk, I wanted to say a few words about an experiment where I ask it, to me, a more important question, which is, can we understand what happens now when the animal is learning from new temporal experiences? So remember that so far, I only considered a case where the animal knows the two distributions, the short and the long, and is flexibly switching between the two. Now, the question is, what would happen if we play with this task and we change the distribution without warning the animal and forcing him to adjust its expectation? So this is exactly what I tried in another experiment, where I first exposed the animal to a fixed distribution, here called the pre-distribution with 5 different intervals. And then at some point in the session, I transitioned, I made a switch to only presenting the longest interval in this case. The reason why I chose the longest interval is because I expected the animal to be highly biased for this particular interval during the precondition. However, after the learning, after the animal has updated its expectations, I predicted that there should be no biases because now the animal should be able to reproduce that specific interval perfectly. In other words, this design allowed me to have a behavioral marker of the adaptation. So here is the behavioral results for one of the monkey who underwent this adaptation experiment. So on the y-axis is the interval that the animal produces. Each single dot is an individual trial. And here on the x-axis is simply the trial number. Here is the data for the pre-distribution where the animal is exposed to the 5 different intervals. And here is what happens when we transition to only presenting the longest. As I told you before, and as we expected, initially, the animal is biased on average toward the mean of the pre-distribution. However, after a few hundreds of trials, you can see that this bias is going away slowly. So the fact that this bias is going away, even though the inputs or the sensory information, the interval here is the same, means that the animal is somehow successfully adapting to the new distribution. So now, obviously, the question was for us to understand and to see whether we could find neural changes that could reflect this adaptation. So here are 6 example neurons from 2 different animals who performed the adaptation experiment. On the x-axis here is the time aligned to the ready. So we are only looking at dynamics during the measurement epoch. And the color code is slightly different. Blue is for the pre-distribution and pink is for after the switch. So as you can see from these 6 example neurons, there is a difference. Several neurons actually change their patterns of activity between the pre and the post condition. So the key question for us was to quantify whether these changes were in line with what we would expect if the animal is updating the expectation. So for that, I will use the exact same analysis that I used in the previous task. In the previous task, we had 2 different distributions each with a different mean, the mu short and the mu long. And I plotted the temporal mapping with the prediction that the slope of this mapping should be equal to the ratio of the 2 means. So now the adaptation experiment, I can do exactly the same. I have the mean before the transition, and I have the new mean after the transition. So now my new prediction is that this temporal mapping should be equal to the ratio of those two values. And to make a long story short, here's the data for one animal. The red line is the prediction and the black is the data. And here are the data side by side for the two monkeys. So as you can see, the speeds, we were able to conclude that the speeds are adjusted according to exactly to the new mean. Now, the last piece of information I wanted to discuss is the fact that we could also quantify the time scale of this neural adaptation. So we did that by quantifying the slope of the temporal mapping, not in the entire post-switch epoch, but instead working in a small running window of only hundreds of trials. So for each of those windows, I could compute the slope and see how it changes during adaptation. So the expectation was that the slope, which I'm going to call lambda, should be 1 before the transition. And then after the transition should at some point converge to the predicted value, here shown by the red line. So this is the data for one monkey. And what we observed is that the adaptation, the neural changes, happened very quickly, in fact, a few hundred trials after the switch. And what was even more remarkable is that when we looked at the behavior, we compared the behavior at this early stage when the neural changes have happened and compared it to the behavior before the adaptation, we did not find any behavioral difference yet, which implies that the neural adaptation precedes to a large extent the behavioral changes that we see. Okay, so now to conclude, I started with two questions. The first one was, how does prior experience in the time domain influence behavior? What I showed is that the monkeys tend to bias their timing behavior towards their prior expectations. I showed that these biases can be captured by the Bayesian theory. And I also showed that monkeys can adapt their expectations in a dynamic environment. The second question was to understand how temporal expectations are represented in the brain. So I showed that temporal expectations constrain the neural dynamics in the frontal cortex, that Bayesian priors, at least in the time domain, create a warping of neural representations. I also showed that the expected time, the average interval expected, is represented in the speed of neural dynamics, that the speed adjustments allow each interval to be encoded relative to expectations, which is in line with the idea of predictive processing. And finally, I showed that during adaptation, the neural adjustments precede behavioral changes to a large extent. So just to finish, I want to acknowledge that this work was done with my supervisor, Merda Jazieri, and also with the help of Hansam Zhang, who is a postdoc in the lab at MIT. And if you have any detailed questions, I can take it now, but feel also free to look at the two papers. They have much more information that I was able to convey today. Thank you very much.
All right. So thanks a lot, Nicolas. Not only was that a very nice talk, but that's also a very beautiful piece of science. Honestly, I really invite everyone in the audience to read these papers. To the audience, please feel free to ask any question in the chat. We still have some time to answer them. Maybe I will just start with a question about your control experiment, in which you are trying to control whether the cortical activity might in fact be encoding the motor preparation.
Right.
So that might be a naive question because I'm not familiar with this experimental setup. But according to you, what would happen if you try to keep exactly the same experiment, but you try to modulate randomly at each trial the distance between the center of the screen and the go target, the target towards which the monkey has to make a saccade? Because this distance is randomly distributed, my naive guess would be that it's completely impossible for the monkey to do any motor preparation, and that the only stuff that you could find being encoded in the cortical activity would be just the expected time.
Okay, I think I see what you mean. Let me just go to the slide where I present the task so it's better for everyone. So are you saying that we could sort of vary this distance between the fixation and the target?
Exactly. I was wondering whether this could serve as a control.
So here, I mean, I see what you're trying to do, but I think it's not exactly the issue we were trying to tackle with the control experiments. Here, the idea was not really related to the target itself. It was more related to whether between the ready and the set, is the dynamics somehow polluted by the fact that the animal will be producing either a short or a long interval on average. We know that the dynamics during the set go epoch is modulated by the interval. That was known even before our experiment. But here, because we see also that the speeds are different during the ready set, when the interval itself is not known, we wanted to make sure that these differences were not because the animal was already expecting to produce a long interval or short interval, but were really an effect of the animal expecting the interval itself to be short or long.
Okay.
So it's kind of decoupling the perceptual phase, the sensory phase of the task from the motor phase.
Okay, so it makes sense, yes. Okay. We still don't have any question from the audience. Maybe I just have a very low level and technical question. When you are performing the population dynamics, you're performing some dimensionality reduction. Which technique are you using? Is it simple PCA or factor analysis or something else?
So in fact, I should have mentioned this, but the figure that I showed is using PCA. So the video. But otherwise, most of the analyses are done in the full state space, meaning that I don't apply any dimensional reduction. Because we don't, like the dimensional reduction here was only useful to get some intuition about the geometry. For instance, the curvature that we see, we were able to see it after applying PCA. But then after, when we quantify, when we project the states along the initial condition axis, or when we compute the speeds, everything is done in the full state space.
Oh, okay.
Yeah. So, but it's a good question. I mean, PCA obviously is not ideal. Like if you want to do some quantitative analysis after applying PCA, it might be problematic because, well, I mean, you may already know this, but PCA tends to be biased in the direction that will recover. So factor analysis is usually preferred. But here again, yeah, I didn't use them for quantitative analysis.
Okay, so you were able to perform all the computations without any dimensionality reduction. On average, how many neurons would that encompass?
So on average for the two monkeys, for one monkey was around 400, 500 neurons.
Okay.
So it's pretty high dimensional, but when you do PCA, which is not the best, but you can capture about 75% of the variance with 3 dimensions. So meaning that the dynamics is relatively low dimensional.
Okay, yeah, that was to be expected. Okay, so thanks a lot.
Yeah, thank you. I hope the talk was not, I don't know, like off the topic of usual talks.
There is no off-topic talks because we really try to cover all the spectrum of neuroscience, from the very clinical and experimental to the theoretical and machine learning side. So I'm really happy to have a talk about predictive coding because it's really of interest for the full community of neuroscientists. And once again, that was not only a super interesting talk, but both papers, they're really beautiful piece of science. Being able to have such beautiful results studying dynamics that are usually pretty noisy, that's pretty beautiful.
Well, thanks.
All right. So I think that's the end of today's talk. Thanks again, Nicolas, for the presentation. Thanks a lot to the audience. And you are all welcome to attend our next NeuroMeetup. All the information are on our webpage. So thanks a lot, Nicolas, and see you next time.
Yep. Thank you. Bye-bye.
Thanks a lot. Bye. Bye, everyone.